Good afternoon, everyone. So you can all now go ahead, take a nap after lunch, and just relax. But I'm going to talk to you about 15 minutes about kind of my journey I've been on for a number of years. And it started back in 2007 when I joined the Moffitt Cancer Center and got into a thing called personalized medicine. So today we talk about precision medicine. Personalized kind of went out the window, and if you are an academic person and you write grants, you know you use the latest buzzword, NIH, or the latest definition, which is the approach for disease treatment and prevention that takes into account individual variability in genes, environment, lifestyle, and in each person. We certainly understand if we take this approach of evidence-based generation as well as evidence-based medicine in the future, that we are going to have steady progress with using multiple omics data, as we've heard today. But it's more than just about the omics data. It's environmental data, behavioral data, other data that we now have the ability to get our hands on very easily, well, not sometimes that easily, but certainly may play a role in the patient's trajectory. We also want to make sure that we're standardizing the data. I wish I lived in a world that if I used a different aligner, I would get the same result. Unfortunately, we don't have that. Or I use a different variant collar, and I get a different result. So we really have to figure out how are we going to standardize data. Do we have algorithms that are tried and true and are reproducible? And we really want to be able to support tools that basically provide new insights into treatments. But when we think about doing this, when I started in 2007, this was pretty easy. We had two things we had to worry about. We had clinical data, which of course is not that easy even today, and we had expression data. But today, if you think about precision medicine is moving way beyond single modalities. We're really looking at how are we going to assign treatment and therapeutics to patients based on multiple omics data as well as other data that I mentioned earlier. And now today, we have the ability of not just to think of diseases as disease. Think about cancer. In the 1990s, we thought of cancer as very much a single disease. And then Chuck Peru, who is now at the University of North Carolina, wrote a seminal paper using expression analysis where he was able to subphenotype breast cancer into 5 different diseases. We want to be able to do the same thing because if we're going to get the best targets for therapy, and we're going to design that therapy for those specific targets, we have to know the exact molecular mechanisms of disease. The way we do that is by stratifying on both the phenotype and the genotype or other omics data to really understand what is that mechanism so that we can distinguish the diseases. So cancer is not one disease, asthma is not one disease, but they are very distinct diseases based on various characteristics. So this is really the goal of what we're trying to do, and really trying to make this happen over a broad range of diseases. Now, we used to think about learning from the individual, but really what we want to think about is how are we going to translate what we learn from individuals as we are going through precision medicine and then make that available to a larger group. So it's kind of taking the paradigm and really shifting it from many to one to many. And why are we trying to do that? Well, if we try to individualize healthcare for every individual, it's unscalable when it comes to a pay scale. I mean, it's just we spend a lot more in healthcare in the United States today than other countries, but when you look at our outcomes, they're no better or worse than many of the other countries that are paying less. So we have to think about how do we translate things into larger groups of people. We know that scientific and technological advances are continuing to grow and at a rapid pace. So now bringing in things like transcriptomics data, proteomics data, epigenomics data, where we can look at a system. Again, it used to be a dirty word, system biology. I think it's coming back finally. Really looking at the system instead of looking at the individual components. This is how we're really going to make progress in this area. And then, of course, we have to look at how will precision medicine harness healthcare and really change that gap between where we are today and where we'd like to go. So when I was presented with this new paradigm, a couple of things happened. One, I started working for a pharmaceutical company, and they decided they were going to sequence 500,000 samples from their clinical trial biobank, plus partner with other groups, such as Genomics England, the UK Biobank, so that now we'll have 2 million sequences. And the first thing I'm like is, how am I going to deal with 2 million sequences? How am I going to democratize this across the company? Not just so that a bioinformaticist can use it, or a data scientist, but that anyone in the company that really wants to be able to use this data has the ability to use it. So it was really lucky when we made this announcement, we started looking at infrastructure, and one of the first deals we made was a deal to manage our genomics data, and we partnered with DNAnexus. And so this provided the foundation for what I was looking for. Number one, it was a platform that could hold not just genomics data, but really could hold multi-omics data. And we could set up standardized pipelines easily within the tool that allowed us to get standardized data that we could analyze with the phenotypic data. We wanted to make sure that we had provenance and auditing, so we knew what happened to that data. Especially within a big pharma, you really want to know who's looking at it and what they're doing with it. You want to be able to transform that data into information, and it had to be scalable. When I used to do GWAS studies back in my day, yes, we had HPC clusters. They ran for a couple of days or weeks. We weren't too worried about it, but we were running 2,000 or 4,000 samples at a time. Today, we have the ability to run hundreds of thousands of samples at a time, as Will was mentioning earlier today. So we have to have a scalable platform, but it has to be wrapped in a secure environment. So this gave me the foundation of what I was looking for. So I had some conversations with some folks at DNAnexus, and we basically came up with we needed to go to the next step. It wasn't just about molecular data. If we were going to make the most benefit from the molecular data that we were collecting, we had to start collecting phenotypic information. And so this was really the goal of creating an integrated molecular precision medicine hub, where, again, we're converting data into knowledge. We're creating knowledge bases based on the data we bring in and the results that we gain. We're really looking at boosting R&D pipeline delivery, so increasing the throughput that we already are doing, as well as then adopting data-driven hypotheses. I mean, this is discovery science, and you have to basically look for things and patterns as discovery, but then you have to actually use true tried science to actually validate them. So when I think about precision medicine, we thought about, well, we need some partners, because DNAnexus, we don't have any clinical data. Okay? It's all of our partners that we work with. And if you think that this is not a global phenomenon, this really is. I couldn't fill up a slide with every single company and organization across the world, so I just chose a few. But you can see that, all of us, a 1 million person plus cohort, and others like Geisinger and Regeneron are quite well known for what they've been doing in this space. So DNAnexus has chosen to partner with 3 organizations. One is Sutter Health, where we're working with them to collect phenotypic data on multiple sclerosis patients, and you're going to be hearing from Greg shortly about that. We are collecting blood and also doing whole exome sequencing on those cohorts. We went ahead and partnered with City of Hope in an oncology project, and working with them to create a precision oncology platform, so that it's not just for City of Hope, but also goes beyond City of Hope to partners that they hope to bring on. And then we also partner with Orion, another oncology network that is run by a company called M2Gen out of Florida. And again, ability to get phenotypic data as well as genotypic data. So we're very excited about these partnerships. But in addition to that, we've already partnered with the UK Biobank and Regeneron basically to make that data deliverable to the partners that are involved with that project. What was the result of all of this? Apollo. So I know you've probably heard about it a little bit. I'm going to tell you a little bit more about it. This was the democratization tool. This was the way to get data into the hands of multiple types of scientists and allow them to look at this data. What changed was this ability. When I was doing this back at Moffitt Cancer Center, I had one database for molecular data, I had one database for phenotypic data. Why? Because it's the only way I could do it. Okay? And what did we end up doing? We ended up passing identifiers around. Today, we have the technology and the scalability because of cloud computing to change that paradigm and bring all of these data together and use them in analysis very seamlessly. So one of the things it allows you to do is hypothesis-driven and exploratory research. We have GUIs for the non-computer people in the world. That's mainly biologists like me who were told, you don't want to take that hard science stuff because that's too difficult. But I don't use GUIs anymore. I'm a bioinformaticist, so I can use backends like Jupyter Lab. But certainly, having this ability for people to explore this data in real time and look at it the way they want to look at it, they can customize the views, is extremely important. We can now start bringing together real-world clinical data and phenotypic data, as well as all of the multi-omics data, imaging data, and other types. And really what we're hoping this allows us to do, and our partners, our researchers at Sutter Health and at City of Hope, is to start really understanding the biological basis of these diseases. There's very little known about the genetic underpinnings of MS. I mean, there's certainly some causative SNPs that are known, but understanding the true mechanisms of the disease is still elusive, and this is something we hope to be able to tease out over the next several years. We do have out-of-the-box apps. So again, it allows people to do analysis with tried and true algorithms that are packaged within the platform so that you can now start not only doing the analysis, but trusting the analysis you're doing because you understand the algorithms that are being used. And with our GUI, it's very easy to put the pipelines together and make them run. As I mentioned earlier, we have integrated Jupyter Lab. This also allows the bioinformatician to bring in Docker or to use R within our platform through Jupyter Lab and really provide that rich bioinformatics experience that most informaticians are looking for. So what we basically created was a new platform that we hope is really going to be a basis for precision medicine that will really help bring together the disparate data that we've been trying to bring together for years or decades. Do it in a way where we do have some semantic harmonization, we do think about metadata and how things are searchable, but also think about the fair data principles. How are we going to start thinking about adopting those ideas and making them a reality? And certainly, that is one of the things that we are going to be continuing to explore to make the data more interoperable and more reusable within our platform. The final piece is, once you do all of this, there's actually kind of some people would call it an upside, some people would call it a downside. It really depends. But when you create these data sets, they become of interest to other organizations. So an academic medical center or a cancer center really can now create a commodity with their data. And this can become very valuable because big pharma doesn't necessarily have access to real-world evidence, and especially real-world evidence on patients that are alive today and getting care today. And so, this is something else that is, right now within the precision medicine space. How can we not only make this data available within the healthcare providers themselves, but how can we partner with pharma and biotech and diagnostic companies to help realize the realization of precision medicine in the future? And I think this is a really hot thing in the near future, and I think will continue to grow. Because it was interesting, I was asked one time, is 2 million sequences going to be enough? And it was at a conference, and I was like, I never really thought about it. But you know what? For very rare diseases and for very rare cancers, maybe 2 million isn't enough. So the only way we really get to where we need to is through partnering, through sharing data, and through using tools like Apollo to really make that data more accessible and more usable to the community. So thank you very much, and I guess we'll take questions at the panel.
Great.